Hello and welcome to Chapter 4. Chapter 4 is the first lecture that we have in this class that really can become overwhelming to students. With the first ones, history, chemistry of life, and the cell, we really didn't go into a lot of different types of analysis. We didn't have to analyze how something worked. We didn't have to understand all the different steps of something. But this is the first one where we're going to look at DNA and what does it do. So there's going to be a lot of definitions on this chapter, and especially this micro lecture PowerPoint, because chapter 4 is going to be broken down into 4 different parts. And the first one is going to have most of your vocab. So let's get started. First, we need to become familiar with DNA and its genes. What does it mean to be a DNA? What does it mean to be called a gene? Because if we can't get past that, we're not going to be able to study genetic disorders and how they affect hereditary traits. We're not going to be able to look at color blindness in chapter 16. Cystic fibrosis we already looked at. Diabetes and hemophilia we look at with blood. We look at diabetes with digestion and the pancreas. So we do look at all these in AMP1 and AMP2, but the key to this is what's DNA, what makes it up, and then how do those genes make a protein. So Mendelian genetics is the same thing. He was the monk that worked with pea plants, and he's the one that helped us realize the correlation between chromosomes, behavior, and his laws of hereditary. Now, DNA and RNA. First, what's the structure of DNA? What is it made of? You need to know all the different building blocks of DNA so you can better understand what it is. We're also going to look at DNA and the proteins are organized and how they actually form a chromosome. Because DNA, chromosome, chromatin, all these things that you start learning, they're just words to you. But if you kind of understand how this is just a couple different things, then these proteins are going to organize the DNA in a way for it to make a chromosome. What's the difference between a chromosome and a strand of DNA? We're going to look at it, and hopefully you understand what makes them different. Then we're going to describe the types of RNA when we talk about RNA. Messenger RNA, ribosomal RNA, transfer RNA. These are all specific things that help RNA finish the job of DNA and the genes to make a protein. So first, deoxyribose nucleic acid. So deoxyribose doesn't have the oxygen here, right there next to that O. Ribose has the O. So it has the oxygen right there. That's the only difference. If it's ribose, it has the O. Deoxyribose without oxygen. It's a thread-like molecule, uniform in diameter, but varied in length. Sometimes they're very short, sometimes they're very long. And the reason why sometimes they're very short, sometimes they're very long, is because they're going to be stored in chromosomes, which we'll look at soon. 46 DNA molecules in the nucleus in most human cells. And it says most human cells because the ones that are going to be undergoing meiosis are going to be half that number because you only want to give half your genes onto your offspring. Total length is around 2 meters. Average DNA molecules around 2 inches long. And I'll kind of show you why I have a picture of chromosomes to kind of give you an idea. What DNA is made up of? A base, a sugar, and a phosphate group. The base can change. So DNA and other molecules, other nucleic acids are polymer of nucleotides. So one sugar, one phosphate, one nitrogenous base, either pyrimidine or purine. So let's look at what's a pyrimidine and what's a purine. Purines are double ringed, adenine and guanine. So you can see they have 2 rings for adenine, 2 rings for guanine. Pyrimidines are only going to be single. So cytosine and thymine. And uracil is only going to be in RNA. So if you look the ones we have in DNA, AT, C to G, A and T are always going to be going together, and C to G are the ones that are always going to be paired together. And when you have RNA, A is no longer going to be paired to thymine, it'll be paired to uracil. We'll talk about that later when we look just at RNA. So molecular shape is a double helix. So it's a spiral staircase going up. Each side piece is the backbone composed of phosphate groups, alternating with the sugar. So you can see the phosphate group, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's what that means on the backbone. The step-like connections in the middle are going to be pairs of the nitrogenous bases. So G to C or C to G, and A to T or T to A, depending on which one comes first. So those bases are going to be held together by a hydrogen bond. And if you look at this, the A to T are only held together by a double hydrogen bond, and C to G has 3 single hydrogen bonds. And if you remember all the way back to chapter 2, when we talked about hydrogen bonds are pretty weak. You can break them apart pretty quickly. And when we talk about DNA and RNA replication, you have specific enzymes that can get in here and separate the hydrogen bonding and allow the gene to be read. So complementary base pairing means one strand determines the base sequence of another. So I told you A always goes to T, G always goes with C, T always goes with A, and this is a way for me to ask questions. And one of our learning outcomes for this semester is, can you use mathematical equations, and can you understand biological systems and figure out a math problem from them? So if I told you you had 50%, or if you had 25% A's and 25% G's, what would be the rest of the formula? Well, if you have 25 of that, you have 25 of this, 25 of that, 25 of C's. Because A is always going to go to T, C is always going to go to G. In the 1900s, components of DNA were known, sugar, phosphate, and base, but they had no idea what they actually did. It wasn't until the 50s where a scientist by the name of Rosalind Franklin used crystal X-ray diffraction and looked at the DNA and was able to show through X-ray diffraction, because of the way it diffracted the light in that crystal, it was a double helix. So Rosalind Franklin was the first one to show that DNA, its structure, was a double helix. And that was like the first real step that started to prove Watson and Crick's model of the double helix. Wilkinson was an English scientist that worked with Rosalind Franklin originally, and Wilkinson was able to write a paper about the mathematics that proved Watson and Crick's model. Wilkins did the math. Rosalind Franklin did the picture that proved the double helix. The 3 men were able to win the Nobel Prize, but Rosalind Franklin died before she was able to receive her Nobel Prize. She died of cancer because most of the early pioneers in X-ray and X-ray diffraction and anything dealing with radioactivity, they died at an early age because they didn't know how deadly that actually could be. So genes, genome, they all look the same. So if we're talking about genes with an S, this is the genetic instruction for the synthesis of proteins. A gene is a segment of DNA that codes for a specific protein. So if you talk about a gene, you're going to be talking about a protein. If you talk about genes, these are multiple areas that can do multiple proteins. A genome is all the genes in one person. In the 1990s, at the end of 1990s, in 1999, they did a project called the Human Genome Project, and it was able to map the entire human genome. And what they did is they took all the DNA, all the chromosomes, and they're able to say there's an A, there's a T, there's a G, a G, a G, a C. It was tedious work, but they looked at each base and they're able to map all of the human genome. 2% of total DNA is part of the genome. The other 98 of our genome is non-coding DNA. They don't really know what it does. They don't know if it's part of, maybe molecular function. Maybe it's part of evolution. They originally called it junk DNA, but they found out some of that junk DNA isn't actually junk after all. Some of that, over the last 25 years, they discovered some of that original DNA that they thought was junk actually goes back into the nucleus and helps maintain and actually helps to regulate gene transmission, RNA or DNA, and synthesis and all this other stuff. So we'll talk about what non-coding and coding parts of DNA and RNA are. So okay, now we get confused and we get bogged down with vocab. Chromatin. These are fine filamentous DNA material complex with protein. What does that actually mean? Chromatin means that if you take the double helix, you then wrap that around a molecule. That molecule is going to be wound around a particle of protein. Then that gets wound around. That gets wound around until it becomes very tightly formed spirals called chromatin. So complex with proteins. We're going to talk about different types of proteins, but pretty much if you took a string and you start spinning it at both ends, and you can see that it starts to get wound up, then the more you do it, that starts to get wound up, until at the end you just have a little ball that's all kind of wound up. That's going to be your chromosome. You have a centromere in the middle that connects chromosomes together. But for the most part, if you kind of look right here, you just have all these little wounding things. All that's going to be parts of chromatin that eventually will make up a chromosome. If you don't quite understand it yet, I do have a couple more slides that are going to go over it, but this is key to understand how DNA will eventually form a chromosome, because a lot of the confusion comes because chromosomes are tricky. So DNA is always in the nucleus. It stores the instruction for protein. Strands of nucleus coiled, allowing much of it to be packed in a small space. Wrapped around a histone molecule, forming a nucleosome. So there we go. We have the DNA. That little circle thing is a protein called histone that allows that to wrap around. These histones, right, becomes a nucleosome, one nucleosome, and now these nucleosomes will get wrapped up and around, and eventually loosely coiled chromatin in non-dividing cells, tightly coiled chromosomes in dividing cells. So chromatin is when they're just kind of loosely going around, and they have to be loose because if it's not dividing, it can have all this different space for all of the stuff to kind of move around. But when it starts to divide, it becomes a lot tighter space, and we'll get into that when we look at DNA synthesis and RNA transcription and translation. So first, now we're going to be during cell division. We have to start becoming tighter and more complex. And when it becomes more tighter and more complex, it starts to form a chromosome. The reason why a chromosome is so tight is because you need it to be tight and in a confined space, because you want those to be pulled apart into different sides of the cell. They're going to be held together in the middle by a centromere. So 2 copies of each chromosome held together at the centromere, which means that's one chromosome, that's one chromosome, that's a pair of chromosomes. So there's 2 there. That's important. 23 paired chromosomes in somatic or general body cells. 23 paired means there's 46 total chromosomes. 46 chromosomes mean you have 46 strands of DNA. So if that made sense to you, carry on to the next slide. If that did not make sense to you, how 23 pairs of chromosomes equals 46 single chromosomes, and those 46 single chromosomes are single strands of DNA that just happen to be supercoiled around one another. So okay, the histones were the disc-shaped cluster of 8 proteins. The nucleosome is going to consist of a core particle, the linker DNA, which is a short segment of DNA connecting to core particles, and then a third shorter than DNA. It's a third shorter than DNA alone. A nucleosome is just when histone gets wrapped around with DNA, it becomes a small ball of nucleosome. Then chromatin protein complex, irregular loops and coils. Now it's much, much, much shorter than the original molecule. So chromatin and chromosomes. Each chromosome is packed in its own spheroid region in the nucleus. Chromosome territory. So in the nucleus, you're going to have chromosomes. They have channels that allow regular chemicals to have access to the genes. Those little nuclear pores allow things to come in and come out. In non-dividing cells, the chromatin is not a static structure. It kind of moves around. And they're calling it chromatin because it's not as tightly packed. It changes moment to moment according to genetic activity of the cell. It's still, even in non-dividing cells, there's still going to be use for the DNA because the DNA is going to have to make hormones. It's going to have to make proteins, enzymes. All these things come from the genes. Genes can get turned on and turned off. You have certain signals that can go into the nuclear pore and have a gene be activated or have a gene be turned off. And chromosomes migrate as cells develop, moving active genes on different chromosomes closer together, allowing genes to partner to bring about developmental tasks in the cell. All that means is that in this area, when you have to have genes being made, a little part unwinds. And that little part is red, that gene gets made. When it's dividing, exact copies are made of all the nuclear DNA. And each chromosome consists of 2 parallel filaments of identical DNA. That's why they're called sister chromatids. They're identical, and they're held together at the centromere, and the kinetochore is the little things on the outside that kind of pinch it together. And the kinetochore becomes important when we have the spindle fibers in mitosis. And it's just protein plaques on either side of the centromere. And we'll talk about the kinetochore when we talk about cell division. So what are the 3 different types of RNA? RNA is much smaller, has fewer bases. Instead of using thymine, it uses uracil. You have messenger RNA. Messenger RNA is the one that gets read from the DNA and then leaves the nucleus. Ribosomal RNA is found on ribosomes, and this is what's going to be reading the messenger RNA to make sure that each amino acid is put in properly. And transfer RNA is the type of RNA that's bringing amino acids to the messenger RNA and the ribosomal RNA to be added together by a peptide bond. RNA is just one nucleotide, not a double helix. Ribose is replaces deoxyribose, so it just removes a sugar, removes an oxygen. And uracil replaces thiamine. So what do they do? What are their actions? We're going to give a working definition to gene and explain why new discoveries in genetics have changed our concept of what a gene is. We're going to explain what a human genome is and what its relationship to health sciences. And we're going to define the term genetic code. What's the process of assembling amino acids to form a protein? Explain what happens to a protein after its amino acid sequence has been synthesized. And then how can we turn a gene on or shut it off? And also, indirectly, how DNA is going to work with non-protein molecules. And DNA only makes proteins. It's your rough, your smooth, your Golgi apparatus, and your Golgi complex that's going to take those proteins and make them into other things. So the previous definition of a gene, just a segment of DNA that carries out code for a particular protein. That's what it used to be. The body has millions of proteins, but only around 20-ish thousand genes. I know it says 25, but there are around 20,000 genes. That means a small percentage of genes produce only RNA molecules, and some segments of DNA belong to two different genes. That was what we previously thought in the 90s. Now we're looking at a gene as an information-containing segment of DNA that codes for production of molecules of RNA that plays a role in synthesizing one or many proteins. Because one gene can have multiple regions on it, we're going to talk about terms called introns and exons. We're going to see how they work together. How does an intron stay on the in, and how does exons exit the nucleus? We're going to take a look at that project because that has changed our reasoning, because in the 1990s we thought there was hundreds or millions of genes. When they did the Human Genome Project, they found out that there was only around 20,000. And the amino acid sequence of a protein is determined by the A's, the G's, the C, the G, the G, the C of the DNA. So the genome, all in the DNA, in one 23 chromosome set. Could be on one side, could be on one of the other sides of the sister chromatids. 46 human chromosomes come in two sets of 23. Each chromosome is a strand of DNA. So one set of 23 chromosomes come from each parent. So when you have meiosis, when you have non-dividing cells in the sex glands for the sperm or the egg from the parents, they're only going to have 23 chromosomes. They're not going to have 23 pairs or a set of 23 chromosomes. So that's what makes you unique. You have 23 chromosomes from your mom, 23 chromosomes from your dad, and then they become your 23 pair of chromosomes later on when the cells have become fertilized. We talked about the Human Genome Project in the 90s, and around 99 is when they started coming up with all the information, and it showed about 99% of the human genome. It started a whole study of genomics, and that's the comprehensive study of a whole genome and how the genes and non-coding DNA interact to affect the structure and function of a whole organism. This kind of changed everything. It started to look at different parts of the body, and even though the body can make millions of proteins, they all come from the same 20 amino acids, and they're all encoded by the same bases, A, T, C, or G. And your genetic code is that system that enables these 4 nucleotides to code for an amino acid sequence of every single protein in your body, over the millions of proteins that you have. So a couple of terms we're going to go over. A base triplet. That means you have 3 bases. It can be AAA, it can be AGC, and then this little base triplet is that unique 3 DNA nucleotide strand that codes for one particular amino acid. The opposite of that is called the codon. The codon is the 3 base sequence on the messenger RNA. So base triplet is on DNA. Codon is the 3 base sequence when it becomes messenger RNA. You have 64 possible codons available to represent the 20 amino acids. This means 61 are going to code for the 20 amino acids. Some are going to be stop codons, which means stop the genetic copying. Some of them are going to be start the genetic copying. And if we're able to understand the basic knowledge of genomes, then we can start making predictions and diagnosis and treatment of diseases. I know it probably doesn't seem like we've gone too far, but since the 90s, we now have 4 or 5 different types of gene therapies that work. The first type of gene therapy that worked was a disease that caused blindness in the eye. And what happened in the eye is that you didn't have a gene to produce an enzyme that broke down a waste product. That built up, you became blind. And I think it was 2008 or 2009, they started doing this gene therapy. And by the time 2017 came along, they were able to create a virus that had that gene in it, and they were able to successfully implant that virus into the eye and the gene into the genome of patients, and they're able to produce that protein. And at an early age, they never lost their ability to see. That has come into other different types of gene therapy. They're looking at cancer, Alzheimer's, schizophrenia, obesity, AIDS, tuberculosis, but these are all very complicated. There are many different types of genes that are involved in these diseases. So just like with the COVID vaccine that was gene therapy, something that's more complicated is going to take longer for us to try to figure out. And by knowledge, early detection of diseases. So if you're missing this gene on this one particular eye disease, if they detect it, they can just give you gene therapy when you're a baby. You'll never know you had it, and you just won't go blind. It also expands potential gene substitution therapy, different types of diseases like cystic fibrosis. These are all ways for us to kind of figure out how we can use genes to help us out. And finally, before we go on to the next micro lecture, how many single DNA strands does a cell have when non-dividing? So it's not dividing. There is 46 single strands. How many does it have when it is dividing and after the DNA has been synthesized? This is tricky. So if you have 46, what's double 46? That's how many it has. Another way to kind of think, how does DNA relate to a chromosome? So if you have 46 strands of DNA, you have 46 single chromosomes. Also, chromosomes are talked about in pairs. So if you have 46 single chromosomes, you have 23 pairs of chromosomes. And chromosomes are in dividing cells, and chromatin is in non-dividing cells. If you can kind of understand everything we just talked about here and how it related to the lecture, then you're ready to move on to part 2.